Welcome to Lesson 6. Data visualization and data presentation are the foundation of the collective scientific knowledge, as our understanding of the set of data is usually limited to what the authors present in their papers, posters, reports, or slides. That's why we will now talk about how to best present our data and any issues related to this topic. Figures and pictures in publications and scientific reports are important because they give authors the opportunity to display a large amount of data very quickly and in a format that is convenient for their readers and for their peers to digest. However, a question that we often forget to answer is, are our figures indeed worth 1,000 words? Or do we actually fail to provide a fair and complete presentation of all data when creating graphs and figures? Let's focus first on bar graphs, as this type of data visualization technique is the most commonly used one in scientific publications for presenting continuous data according to the analysis of several hundreds of research articles published. In a study published in 2015, it was found that over 85% of publications in psychology journals between January and March 2014 included at least one figure with a bar graph. It is worth reminding ourselves that bar graphs are designed for categorical variables that contain a finite number of categories or distinct groups like dead, alive, or obese, overweight, normal, underweight. However, they are commonly used to present continuous data that can have any value between a theoretical minimum and maximum, like birth weight, temperature, neutrophil count, etc. So bar graphs of continuous data typically show the mean and standard error of the mean, and we will now focus on why presenting the means and the standard error of the mean of continuous data as a bar graph or in a table format is actually problematic in a lot of cases. Let's take an example of a study that involved two groups, a control and an experimental group. According to the currently dominating but not ideal practice, the study outcome may be presented as two bars, one bar representing mean and SEM for one group. The problem is that we often ignore the fact that many different data distributions can lead to the same bar or line graph as shown on this slide. For example, summarizing the data as mean and SEM may cause readers to wrongly infer that the data are normally distributed, like in panel B. But it could also be that outliers exist, like in panel C, or that different subpopulations might be present, like in panel D. Or it could be critical for the reader to understand whether different sample sizes have been used for the different treatment groups, as shown in panel E. All this information is lost or hidden by choosing bar graphs instead of more meaningful and appropriate alternatives. Even more problems arise when bar graphs are used to show paired or non-independent data. Bar graphs of paired data wrongly suggest that the groups being compared are independent and provide no information about whether changes are consistent across subjects or experimental units. For example, in panel B, values for every subject are actually higher in the second condition. In panel C, there are no consistent differences between the two conditions. And panel D actually suggests that there may be distinct subgroups of responders and non-responders. So only scatter plots provide a clear picture and demonstrate whether or not differences between two conditions are consistent for each subject. In contrast to bar graphs, alternatives like scatter plots or box plots allow readers to properly examine the data distribution, are more informative than the mean and the standard error of the mean, and represent legitimate forms of how data should be presented. These alternatives enhance readers' understanding of the data while also allowing readers to detect violations of any statistical assumptions. The increased flexibility of scatterplots also allows authors to convey study design information. For example, in small sample size studies, scatterplots can easily be modified to differentiate between datasets that include independent groups and those that include paired or matched data. Overall, while scatter or box plots prompts the reader to critically evaluate the author's analysis and interpretation of the data, bar graphs discourage the reader from thinking about these issues by masking distributional information. It is important to note that also the scaling of the y-axis can cause bias in the perception and interpretation of the data presented. In this example, the graph uses a y-axis starting at 80% and creates an impression that there is a meaningful difference between the two groups. If the same data are plotted using a neutral y-axis starting at 0, the difference between groups becomes less obvious, and it can be questioned whether there's actually a biologically meaningful difference between the two groups at all. Therefore, as a rule of thumb, axis scaling should cover the full range of the possible data, and the scale should be biologically or clinically meaningful. The default should be to start at 0 for all ratio variables. Of course, exceptions from this rule can be absolutely adequate, for instance, when displaying mammalian body temperature, heart rate, or blood pressure. But in many less obvious cases, authors should transparently explain and justify why axis scaling does not start at 0. When presenting our data, we also need to consider the choice of depicting data variability, as this, at least psychologically, affects how the dataset is perceived. So one key question is, which type of error bars is scientifically most appropriate for my study or my experiment? The example shown here has explored possible differences in relaxation of the urinary bladder by several beta-adrenal receptor agonists between young and old rats. By looking at the scatter plot, we notice that there was one rat amongst the old rats in which noradrenaline caused hardly any relaxation. It does not look like a major outlier, but likely had impact on the overall mean. Second, there's considerable overlap in the noradrenaline effects between the two age groups. So only 6 out of 9 measurements in old rats yield values smaller than the lowest value in the young rats. However, the graph with means and SEM error bars makes it look as if the difference between the two groups is quite robust, as the between group mean difference is about 3 times larger than the magnitude of the error bar. But as we have discussed before, this is not what the data really say. On the other hand, the standard deviation error bars by definition are larger. About 95% of all data fall within 2 SDs. Looking at the SD error bars, it is quite clear that the two groups overlap. This is what the raw data say, but not the impression coming from the SEM error bars. So, SEM, which is the SD divided by square root of n, has the smallest error bar, and a small error bar may make even small group differences look large, even if the overlap between both groups is considerable. There is also a conceptual difference between SD and SEM error bars. SD describes the variability within the sample, whereas SEM describes the precision with which the group mean has been estimated. An alternative to presenting precision of the parameter estimate is the 95% confidence interval. In this specific case, it provides a similar message as the SD error bar, in that the two populations may differ but probably are overlapping. Finally, for data from populations with non-Gaussian distribution, medians with interquartile ranges are the preferred option to present data when scatter plots are not possible. In summary, many datasets may lead to similar bar graphs, but a different biology may be hidden behind it in each case. Therefore, the scatter plot where possible, is clearly the preferred option of showing quantitative data. If means with error bar have to be shown, for example, within the main text of a publication, SD is the error bar of choice to depict variability and confidence interval to depict precision of parameter estimate. Let's focus for a minute on the concept of normalization and any potential consequences attached to the normalization approach. Quite often, datasets are presented after normalization for some other variable. For example, by using reference controls like housekeeping genes for PCR or Western blotting-based experiments. This often makes sense and can be scientifically justified, but there are certain risks to be considered. In the example shown here, the abundance of several G protein alpha subunits were measured at the protein level. First, without using GAPDH as a loading control for normalization, as shown here. For the GS and the GI1 phases, it looks like the expression of G protein alpha subunits was reduced, in contrast to the GI2 phase. After that, data were then normalized for GAPDH, as shown here in the bottom row. And what you can see is that normalization for GAPDH indeed changed the conclusion of the study, as the reduction in GS and GY1 vanished, and the lack of change in GI2 looked now more like an increase. So why is that? The explanation is that the GAPDH expression actually changed upon isoprenaline treatment and did not stay at the same level. In fact, for the experiment shown here, a 32% lower expression of the GAPDH protein in cells treated with the beta adrenoceptor agonist was detected compared to the control samples. So in this case, given that the GAPDH expression is not stable under the experimental conditions, GAPDH is indeed not suitable for normalization of this immunoblot data. So therefore, other alternatives for loading controls should be considered. For example, carefully measuring the whole protein concentration in the samples generated. And this may be more appropriate to actually avoid misleading interpretation of the results obtained. As a general guidance, if you present normalized data, explain exactly how you did it and how you have defined the 100% and the 0% ranges for your specific experiment, as this has a direct influence on the conclusion of your study, as we have seen in the example before. To summarize, normalization may be misleading if the denominator changes, like in the GAPDH example, and normalization makes only sense if the denominator remains stable during the experiment. Studies are typically underpowered to detect significant denominator changes. It is highly advisable to report quantitatively what the denominator was in all groups. We should carefully consider whether normalization actually makes sense for our specific experiment. And even though normalization is done frequently, we still need to think about any potential implication and risk factors. And finally, it is important to pre-specify the normalization procedure in our study protocols. The last topic of this session about data presentation, which we would like to discuss, is related to choosing the most adequate statistical method, which affects the way how you analyze as well as present your obtained data to potential readers. There are basically two types of statistical inference methods. We can either test hypotheses about experimental parameters, or we can estimate these population parameters. We have talked about hypothesis significance testing before, and we will now introduce the estimation approach to inference process and compare it to the NHST method. Currently, most scientists approach inference through null hypothesis significance testing. Using the null hypothesis testing approach, we would ask a qualitative question. For example, does a certain drug influence learning? The data collected is then reduced down to a test statistic and a p-value, and from these we make a qualitative conclusion. Yes, this drug influences learning. Results are often treated as definitive, so there can be little motivation to actually conduct any replications. Why test the drug again when it has been shown to work? In contrast, the estimation approach to inference reports effect sizes with expressions of uncertainty. The estimation approach can help to avoid overconfident claims from inadequate samples, can improve comparisons of results across context, and can provide a straightforward approach for planning informative studies. The estimation approach involves a shift in how research questions are conceptualized, designed, and reported. Rather than a qualitative question, we ask a quantitative question. How much does this drug influence learning? This is answered with an effect size, which gives a quantitative answer to the research question, and an interval estimate, which helps express uncertainty. For example, the drug in this example improved memory by 10% with a 95% margin of error of 9%. The effect size expresses the magnitude of difference observed in the sample, here 10%. And the magnitude of error expresses some of the uncertainty in generalizing to the population. In general, the common theme of the estimation approach is an emphasis on uncertainty, which is a key aspect of good statistical practice. Focusing on uncertainty makes the tentative nature of any one study more salient and highlights the need for direct replication. To highlight the contrast between testing and estimation, let's summarize the same data with both approaches. As an example, you have seen the recent report in Nature Neuroscience examining the effect of caffeine on memory, which is part of the essential reading list for this lesson. So for this study, participants were asked to memorize different images and then got either caffeine or a placebo control. And the next day, participants were then questioned about what they remembered about the pictures shown on the day before. In the original report, results were summarized using hypothesis testing, with a t-test indicating a statistically significant enhancement in memory in the caffeine group relative to the placebo group as shown here. This figure shows how such data are often presented. A bar graph with error bars representing SEs and an asterisk to denote a statistically significant difference. And from this, it was concluded caffeine enhances memory consolidation in humans. Now, the estimation approach provides a different lens for interpreting the same data. In this approach, we would ask a quantitative question. To what extent does caffeine improve memory? To answer this question, we estimate the difference between the group means and then quantify uncertainty in this estimate due to expected sampling error. The outcome would be that caffeine is estimated to improve memory relative to the placebo group by 31% with a 95% confidence interval of 0.2% to 62%. The figure in panel B graphically represents this information with an estimation plot that shows the effect size in the sample and the confidence interval for the effect. Critically, the confidence interval suggests considerable uncertainty about generalizing from the sample to the world at large. If the real effect were very large, 62% increase, these data would not be especially surprising. Similarly, if the real effect were vanishingly small, 0.2% increase, these data would also not be especially surprising. Moreover, this wide range of possibilities is optimistic, as it is based on uncertainty due only to sampling error, with the assumption that all other sources of error and bias are negligible. Reflecting on this uncertainty makes it clear that although the study is statistically significant, the sample collected is not adequate, yielding a poor signal-to-noise ratio as the margin of error is only fractionally smaller than the observed effect.
This can be classified as low power, and the study yields too much uncertainty to provide a clear answer to the research question. The most appropriate interpretation at this point would thus be caffeine probably does not impair memory consolidation.
So we would like to finish this session by reminding you that the argument "everybody does it" is indeed not necessarily a good argument for doing it ourselves. And by just following what everybody else does might be one of the reasons why it is so difficult to introduce changes to the current scientific environment. Although following good research practice standards should be the most normal thing to do for us scientists.